Hey, good morning, everyone. Just gonna make sure I got this. All right, excellent. Yeah, so today we're gonna be talking about some of the tool sets and frameworks of thinking that we use at Séran as we're going through all of these different API problems that we have. We've heard a lot about them, right? Low solubility, low permeability. We'll talk a little bit about how we classify some of those, how we select technologies, and look at two different case studies. Right, so really quickly, I already got a kind introduction. I'll just introduce Séran. So we're a science-first CDMO out in beautiful Bend, Oregon. If you ever want to come visit, we'd love to say hey, host you, show you around. So science first and data-driven, right? So we really want to not just take a templated approach to what we're doing, right? We want to understand what are the problems and shortcomings of the API that we're working with. And that way we can start to come to better formulation decisions earlier in the process, right? So we want to be able to identify robust, scalable, right, manufacturable products as early as possible. So to kind of illustrate what I mean there is, we'll start by contrast, right? The typical paradigm where we're formulating fit-for-purpose formulations throughout the process, right? So maybe preclinical, your suspensions and solution formulations. First in human, maybe you've got a powder in bottle or a powder in capsule. Well, at some point you get a tablet, maybe you need to reformulate that tablet once or twice more, right? So as you're going throughout the development life cycle of your program, you're having to reformulate, spend a lot of time, a lot of money, doing RBA studies, bioequivalency studies. So it's a lot easier to just get a good drug product from the start, right? So this was motivated by oncology programs, right? Indications with rapid approval timelines. But we really want to take this approach and apply it to every program that we have, right? So ideally, the tablet or the capsule that you take into your first in human study is the exact same formulation that you take through phase 2, phase 3, and into commercial, right? So in order to identify the formulations that early on, we're using a lot of material sparing approaches, right? We want to understand, or we know that, oftentimes when you're coming out of preclinical and into first in human, right, you might not have the most API, right? So what can we do with milligrams? What can we do with grams? And how can we make sure that the formulations we're coming to are representative, right? So we actually heard yesterday some great talks about how particle density and size really affects your final product, not just from a dissolution perspective, but also from a tabletability perspective, right? So these are some of the things, so at Séran we've put a lot of thought into, right? So we have some custom spray dryers that help address a lot of these different scalability challenges that come with the particle engineering of spray dried dispersions. So getting into it a little bit, right? So some of the frameworks that we're using when we're approaching these different programs, right? We need to know a bit about not just the drug itself, the drug substance itself, but the end goal, the target product profile. So, of course, with the API, you need to understand what's its solubility throughout the gastrointestinal tract, not just with relation to pH, but also with relation to bile salts, right? What are the dissolution rate limitations or properties? How permeable is it? Right? And then, of course, stability. We want to make sure that any formulations that we're proposing are compatible with the API and its sensitivities. Right? So we get this information, we measure it, we take whatever you provide, right? And then we also want to combine it with the target product profile, right? So typically with formulations, you're looking at performance, manufacturability, and stability, right? Kind of the traditional three main pillars. We like to add in a fourth one, design, right? We need to understand what your dose is going to be for preclinical, clinical, commercial. What's your patient population, right? Do you need pediatric dosing? Does it need to be compatible with any geriatric populations? Or are there going to be any proton pump inhibitors? Is there anything about your patient population that might affect the physiology of the gastrointestinal tract, right? Often you see exposures change as you go from healthy volunteers into patient populations. We want to try to preempt that as early as possible, right? So you can see we're really trying to incorporate all of these, and then we also want to make sure that if we're using an enabled intermediate, we're thinking about that final drug product as we're formulating the intermediate. There's a lot of synergies that you can utilize when you're making an amorphous solid dispersion and thinking about the tablet that it's going to go into. And conversely, there's some constraints that one might place on the other as well. Right? So we'll look at a couple of these different case studies. And one more framework that we use, and we've heard a lot about the DCS already, so I won't belabor the point, but there's a couple of subtle differences that I wanted to point out between the DCS and the BCS and how we're using that to guide our formulation, right? So BCS is really focused on worst case solubility and USP buffers across a physiologically relevant pH. So it's really designed to help guide regulatory perspectives on bioequivalency studies. The developability classification system, on the other hand, it's more of a risk assessment for limited oral drug absorption based on aqueous solubility in the intestine, right? So it's really focused on where the API is going to be absorbed, right? And that comes with some benefits. It also comes with some caveats that we need to keep in mind, right? So it's not going to be thinking about any kinetic effects that might be happening in the body, right? Any drug that you take orally has to go through your stomach first to get to the intestine. So we'll look at a case study that addresses some of that as well. So I do really like it though. It breaks down, right, like our BCS class 2, now we have 2A and 2B, right? So we can balance not just our solubility, but how's our dissolution rate gonna interplay with that. So if you have a high solubility, but your dose is really high, you may still be in the 2B, solubility limited range, or maybe your permeability is good enough and your dose is low enough that you're able to absorb the drug as it's dissolving, and you never really hit that solubility limit, right? So it kind of sheds some light on where we might need to focus our attention as formulators. All right, so we can take that exact same DCS plot and we can superimpose it over some of our technology choices. And the axes are flipped here, right? So we have log P down on the bottom as our stand-in for permeability. Log P also gives us a little bit of information about which sorts of lipid or emulsion formulations we could turn to, right? And then of course, dose number is on the y-axis. So, right, by understanding where we sit in this formulation space, we can turn to we can turn to different technologies. So, right, if we're in the DCS1, DCS2, right, maybe micronization or nanomilling. We've heard a lot about these technologies today. And then I'll also point out if you need to affect your dissolution rate, amorphous solid dispersions also do that, right? It might be more than you need, right? But you're disrupting the crystal lattice, you're pairing it with a polymer, your dissolution rate's gonna be a lot faster than the ingoing crystalline material. But if you need solubility enhancement, right, there's obviously other technologies. We've heard a lot about lipid systems, amorphous dispersions. There's, even within those options, more options, right? So how do we find the solution that's right for the program? So we're gonna go through 2 case studies today that kind of straddle a couple of different solutions that we could turn to, and I'm gonna walk you through how we're thinking about the final drug product and what we could choose to advance throughout development. All right, so case study 1, we're looking at something that's right on the edge of micronization versus amorphous forms probably being required. And for this program, the final drug product that we ended up with was we call it an IR plus tablet, right? We're including some enabling acidulants and precipitation inhibitors. But how did we get there, right? So the client came to us with a weak base, as you can probably surmise based on the solubility versus pH profile over here, right? So we have really good, tens of mgs per mil solubility and low gastric pH. And then as your pH goes up, your solubility goes down. And by the time we're in intestinal media, right, we're below a mg per mil, below 0.1 mgs per mil solubility. Now, the target dose for this program is a little bit higher, right, 800 milligrams. So based on that and the solubility in the intestine, right, DCS would classify it as 2B, solubility limited absorption. And based on its solubility in the intestine, we would expect there to be little to no exposure based on the DCS, right? But when the client took the API, milled it down and dosed it into the clinic, they actually saw pretty good exposure, but high variability. So they came to us trying to solve the variability. Now, when we see weak base, we see a solubility profile like this, and we hear variability, our thoughts immediately turn to what's going on in the stomach, right? We like to think in the lab that every human is perfectly pH 1.6 in the fasted state, right? But there's a lot of variability, even in healthy humans, between people, within people, right? And just the fasted state. So our hypothesis for this was that variability in dissolution, extent of dissolution in the stomach affected our exposure in the intestine. So first we need to confirm that this is the mechanism at play, and then we need to identify some formulation strategies that get around that and help mitigate these different effects. So one of the experiments we love to do the most is biorelevant dissolution, right? It really gets us lets us probe what's going on a little bit in the different media. We can see what sort of kinetic factors there are, right? So we're starting out, of course, in our simulated gastric fluid and then diluting it into fasted state intestinal fluid. And we took some measurements on this API, right? So we were able to measure its crystalline solubility as well as its amorphous solubility. And as we heard about yesterday, right? Amorphous solubility is going to be the highest amount of free drug that you can have thermodynamically in your media. And so that's going to always be higher than your crystalline solubility. Okay, so let's start with the base case. Right, we take our API, we add our target dose, 800 milligrams, and we put it in 250 milliliters of pH 2 simulated gastric fluid. As you would expect, our entire dose is able to dissolve. And what's interesting is when we then dilute it into our fasted state intestinal media, we're not precipitating all the way down to our crystalline solubility. It's able to amorphously precipitate and then sustain that amorphous solubility throughout the extent of the experiment, right? So we're actually seeing this quite a bit more now. Not everything is a traditional brick dust or spring and parachute rapid crystallizer. We're seeing a lot more, especially with new classes of compounds like protein degraders, molecules that are able to self-sustain their amorphous solubility. All right. So what happens when we have a low or a higher gastric pH in a lower solubility environment? Right, so we're not completely dissolving our dose, and therefore when we're going into our intestinal media, there's a lot of undissolved solids, and we're not able to achieve that amorphous solubility. We're down at our crystalline solubility. So we always like to think of, like, we look at the data and we like to think of it, our mental model, in terms of what's happening in the body. Right, so case one, right, we're in the stomach, we're fully dissolved. When we get to the intestine, we have our amorphous solubility, so high levels of free drug. We have amorphous precipitates, and that gives us a really steep concentration gradient across the intestinal epithelium. So we're really driving diffusion by having, driving absorption and exposure by having our amorphous solubility here. But when we have incomplete dissolution, right, we have not only a lower level of free drug, but we also have crystalline undissolved solids, and we have a shallower concentration gradient, and therefore less of a thermodynamic drive for diffusion across the intestinal epithelium. So based on this model and based on these results, right, the solution is obvious. We need to make sure that we are dosing amorphous material into the intestine. And it doesn't really matter how it becomes amorphous, we just need to get it there, right? So one obvious solution is an amorphous solid dispersion. That's exactly what we did here. We formulated an enteric SDD and put it into our pH 5 dissolution, right? So it's an enteric polymer, so we're not releasing as much API into our gastric media. But then as soon as we get into our intestinal media, where we have the pH 6.8 and the polymer solubilized, we're able to release the drug. And we're able to sustain our amorphous solubility throughout the duration of the experiment. And we even have some formation of drug polymer colloidal species, which is that solubility that we're seeing above the amorphous line. So this approach works, right? Our problem statement was we need amorphous material. We got amorphous material, and we're able to maintain that. But that might be over-engineering a solution. Is there a simpler way? As I alluded to at the start, right, we thought of an IR tablet, IR plus tablet. So what's going on here is as the tablet is disintegrating in the stomach, the acidulant is altering the microenvironment of gastric media around the tablet, right? We're not adjusting the whole stomach, just what's around the tablet and around the API. That drives it into solution, and then we have a precipitation inhibitor in there to help maintain it in that supersaturated state. Now, I mentioned that this API in particular is not a rapid crystallizer, so the precipitation inhibitor may or may not be necessary, but especially for strong crystallizing salts or crystalline forms, a precipitation inhibitor would probably help. So we took this formulation and we put it in our same dissolution experiment, right? High gastric pH, we should be low solubility, but the acidulant is able to drive the API into solution. And once we transfer that fully dissolved API into our intestinal media, you can see we're able to achieve and maintain our amorphous solubility throughout the duration of the experiment. So 2 ways to solve this problem. Which one should we choose? Well, for this program, we chose an IR plus tablet, but for for any other program, right, we want to balance out all of the different pillars, right? So both the amorphous solid dispersion and the IR plus tablets check the box for performance, all right? So what else can we use to eliminate one formulation or select one over the other? Well, stability might be an option, right? A lot of APIs have acid catalyzed hydrolysis or other sensitivities to acids, so an acidulant might be exactly what you don't want in your tablet. It could affect your chemical stability, right? Or it might drive what sort of amorphous solid dispersion you make. If you're choosing an enteric polymer, right, you're gonna have succinic acid and other acidic groups that could affect your chemical stability. So again, that's gonna be dependent on the API. Similarly, manufacturability, right? So let's say your crystal form is large and needle-like and has terrible flow and poor compactability. It may be worthwhile to dissolve it, make an amorphous solid dispersion, and even if it's an STD and you have to do dry granulation, you're gonna have good compressibility and decent powder flow. So that might be worth it for you. Or, right, maybe you don't want as many unit operations as are needed for an amorphous solid dispersion, and you really wanna just try to make the crystalline solid work. Right, so again, balancing the needs of the program. And then finally, design. I'm gonna bring this up because, right, it was a higher dose, 800 milligrams. So an amorphous solid dispersion, depending on the drug loading that you're able to achieve, may not be enabling for this program, right? Depending on the pill burden, if there's any constraints on tablet size for the patient population, right? So, right, these are some of the different questions that we went through when we were identifying which technology to select. For this program, we opted for the IR+. Okay, so let's get into the next case study. So this is actually some research that we conducted. It's on a single formulation, but we're comparing 2 different ways of making it, right? So spray drying and hot melt extrusion. Both mature technologies, both are highly scalable, and both of them producing chemically the same thing, right? This is 2 SEM micrographs of a 25% drug-loaded HPMCASM ASD, right? So chemically they're the same, but I mean, you can clearly see that we're not working with the same material here. STDs, right, you have much lower particle size distributions, so a higher surface area to volume ratio. You're also lower density, right, because you have to have some porosity and your wall thickness, right, as you're diffusing. I believe Nemo was talking through that just yesterday, so I won't dive too much into that. So lower density, higher particle size, or higher surface area to volume ratio. HME, conversely, right, you're producing a single monolithic solid that tends to not have any air trapped in it. And then you're milling it down, right? So you have some control over your surface area to volume ratio, but particularly if you're trying to leverage direct compression tablets, there's only so low that you can go, right? So there's gonna be pros and cons to either of these approaches. So to kind of illustrate these, we turn to itraconazole, well studied, there's a wealth of literature on it, right? So we're not looking to find the optimal formulation here. We're trying to understand how our technology choices affect our final drug product, right? So we're gonna look here at STDs and HMEs that we made of this formulation, and we're gonna compare the powders through biorelevant dissolution, and then we're going to look at the manufacturability and tabletability of those ASDs. So first up, right, same biorelevant dissolution scheme. We have our gastric media being diluted into intestinal media. And right off the bat, you can see there are differences, right? So I will say both ASDs are able to achieve and maintain their amorphous solubility throughout the experiment, which was taken out to 20 hours, right? So based on your program, either of these could be advanceable, right? Some programs we've seen the formation of colloidal species does not directly translate to better exposure. Some of them it doesn't really matter, right? But some of them it is incredibly important, and itraconazole is one of those cases, right? So based on that, right, the STD, we have higher surface area, some surface enrichment, we're able to extract more API in the gastric phase, and then we get to the intestinal phase and we're able to better form those drug polymer colloids that can impact bioavailability. Right, so in this case, right, it looks like the STD might be the winner for this program, but let's take it further into the tablet, right? Let's see what happens, because it's got to get into you somehow, right? So when we're thinking tabletability, there's obviously a lot of different metrics, but the two obvious ones when we're comparing STD to an HME are flow, right, and tabletability. So, right, just as you would expect, the HME has amazing flow, right? We can definitely do a direct compression tablet here. That could be a smooth process. The STD has surprisingly decent flow. It's certainly enough to enable a dry granulation approach, but maybe not enough to enable a direct compression tablet. Now, the other metric that we're thinking about, right, is tabletability. How well are we able to compact and compress and form those interfacial bonds between our granules and particles, right? So there's two thresholds that we think of at Séran. There's a lower threshold, which is based on tensile strength, right? So we need to be able to produce a tablet that is strong enough to survive coating, packaging, transport, storage, sitting in your glove box, right? All of that. So anything below 1.5 to 1.8, right? We typically see that as our cutoff, 1.5 to 1.8 megapascals. And then above that, right, from a hardness perspective, you're more just limited on your dissolution rate or any dissolution specs that you might have. Now, the upper threshold that we think about is more practical. It's just based on our commercial or our larger scale tablet presses, right? We can comfortably get up to 250 megapascals. Anything beyond that, right, the operators start to look at us weird. So we try to keep our compression strength, our compression stresses, below 250, and we want to make sure that we're able to achieve tensile strengths 1.5 or higher, 1.8 or higher. So you can see here, we did slugging and milling, we dry granulated our STD, and we have excellent sensitivity across that region, right? We're able to identify a clear process parameter where we can scalably produce strong tablets. Now, the HME formulation, it's the exact same excipient ratios, direct compression, but because we have larger granules with less surface area and they're denser, we lose out on quite a bit of compactability or compressibility. And we're not able to identify a scalable process. Now, I will say, if we wanted to make the HME work, there's obviously things we could do. We could mill it down lower, we could have to increase our surface area, or we could include a processing aid, right? So we could go back to the formulation board and include, right, some span or some poloxamer, right? So there are strategies we could use to make the HME work, but if we're expecting these to be one for one, not only in our dissolution, but our tabletability, I think we just need to reevaluate that. So when we're making these different decisions on which technologies we're choosing, we need to understand how they're going to affect our particles, because the particle engineering is going to have massive effects, as we've seen, on our performance, manufacturability. Stability we didn't touch on, right? But if you have thermal sensitivities and your process requires you to get a bit hotter, right, then HME might not work. Or back to manufacturability, if you have low organic solubility, you're just not going to be able to make a scalable STD process, right? So we got to balance all of these things. So I think I'm running out of time here, so I'm just going to leave you with these thoughts, right? We want to understand our problem statements, the limitations of our programs, and so that we can identify as early as possible what formulation strategies and technologies we want to utilize throughout the development life cycle here. So there's always going to be multiple answers to any given question. So I've shared some of our toolkits for being able to identify what those answers could be and select which one's best for your program so that you can develop a drug product right from the start. Thank you. I'm happy to answer any questions.
Thank you, Wesley. Obviously, we're going to have a break shortly after this, but any quick questions for Wesley before we break out? I know, Wesley, you've got Jerome and Jaspreet with you as well.
Yep.
So,
We have a booth in the back corner. Feel free to come by and ask us questions.
any final questions? Yes.
Did you observe any differences in?
Sorry.
Did you observe any differences in the disintegration patterns for the melt extruded compressed tablet versus the spray dried dispersion compressed tablet?
Yeah, there were some small differences, but they were all able to disintegrate fully with under 5 minutes. So there were differences, but nothing there were no red flags, I'll say.
And release profile was comparable?
So I didn't show it today. So release profile and sink dissolution we didn't test, and like the release method for itraconazole. But from a biorelevant dissolution perspective, the differences that we saw in just the dissolution of the powders became accentuated in the dissolution of the tablets because you have the additional step now. So the HME, we think, was forming fewer colloidal species because it was slower to be able to dissolve and have the polymer help bring the API into solution. So by adding the additional steps of disintegration and primary dissolution, you're slowing it down even further, and it was barely able to form any colloidal species. Yeah.
Great talk.
Thank you.
There's a perception that HME is sort of the fallback plan when SDDs don't work. What's your response to that? When they're basically the same thing, different forms, but they're SDs, right? But what's your response to that?
I mean, it really just depends on the needs of the program, right? We've had some where an HME was really the only thing that we could get to work because the organic solubility was low enough, right? I think more often than not, we see people want to start out with spray-dried dispersions because you can be a lot more material sparing at first, right? We can do formulation screens with 100 milligrams active or so. And then they want to switch to an HME later on in the program. And then what we end up seeing at time and again is that, the differences in particles makes a difference in the product. So, I don't think one technology is better than the other. Obviously, we do a lot of spray drying at Séran, but it's not the only thing we do. Thank you.
I have a question here. When we are comparing SDD and HME, have you taken into consideration the processing conditions of the HME? Having the same formulation and processing them in different conditions may give you different release profiles. The reason is, are you going to get amorphous domains or molecular dispersion? So have you taken that into consideration?
Yeah, no, absolutely. Neither of these were optimized processes for either the SDD or the HME. So, if we were to do a truly apples-to-apples comparison, like you're suggesting, we would need to, fully optimize the process for the SDD versus the HME. Yeah, you're right. I think that's part of, why we would want to select a technology early enough that we can really focus on optimizing the process as opposed to optimizing the formulation. If that answers your question. Thank you.
Yeah, a wonderful presentation. I can clearly see you're coming from a perspective of someone who has to do it day in and day out. I appreciate that. So just a general question I have, I see you use the acidulant to maintain that pH and get the solubility going on, and that was a good option for you. Have you ever given any thought to having additional flexibility if you just have the acidulant administered separately, say as like a separate tablet or something? Does it still promote the same performance in the microenvironment, or have you seen that that's not necessarily the case? That's only the potential trade-off I could perceive,
Yeah, no, that's a great question. We haven't looked at that directly, but what I would guess is since we're really focused on altering the local environment within the tablet as it's bringing in moisture, that having the acidulant separately, you'd have to include a lot more acidulant so that you're actually modifying the pH of the entire stomach media as opposed to just what's in the tablet.
Yeah, makes sense.
Yeah, thank you.
Hi, great talk. I just had one perspective on the particle size differences and density differences between spray-dried material and HME. I think yesterday we had a similar talk from Microsize on kinetisol, where it was kind of opposite. The kinetisol having higher particle size or higher bulk density gave better solubility. So do you think it's subjective to the molecule, to the formulation, rather than the particle size differences in general?
Yeah, absolutely. I would agree. There's a lot of good literature out there, right, on congruent release of your API and polymer, and making sure that to achieve the formation of drug polymer colloids that you have polymer governed dissolution mechanisms. So, which one is better is definitely going to depend on your solubility and dissolution rate of the API, right? So I've seen cases where having like this, right, where having the higher surface area, lower density particles, you're really able to drive the formation of those. But I've also seen the opposite, right? Like you're suggesting, we're having protection from the gastric media, right? Depending on the solubility of your API, slower to dissolve particles. That ended up being more helpful, right? If you have an API that's wildly soluble in gastric media, you might end up leaching it all away, especially if you have a neutral polymer, right? And that's also going to be soluble, and you might disrupt any interactions that there could be between the drug and the polymer. So it's definitely something that we want to evaluate in vitro on a small scale, because I don't think that's something that, at least yet, right now, we can't predict that beforehand.
Right. Thank you very much, Wesley. Thank you.
Thank you.
Okay.